keynote by Dr. Gesine Lorenz from Hydroisotop. It's going to be a keynote on determination of biogenic carbon content of industrial CO2 emissions. And she's a scientist at Hydroisotop, and they do environmental assessments in the field of groundwater dating, water quality, nuclear waste deposits, geothermal energy, and biogenic carbon of food products and CO2 emissions.
Okay, thank you. I was already introduced, so I can quickly start with my presentation. I'm sorry that I changed the title, but I don't really understand the word assessment, and I think a lot of you know more about the assessment, while I'm more in the expertise of the analytical methods to measure the biogenic carbon content of industrial CO2 emissions. Why do we need analytical methods? Why do you want to measure this renewable part of the carbon emissions? First of all, I guess it's the national reporting of fossil CO2 emissions within the EU or worldwide. Usually you need a better knowledge of fossil and renewable carbon percentage of all materials and emissions. Of course, you can use the analytical results for their CO2 certificates, which several industries have to buy. Then the knowledge is very valuable for your strategies for the reducing of fossil CO2 emissions, and you can use it for advertising, and there's a lot of more reasons why you should quantify the fossil CO2 emissions from the renewable. Why do we need analytical methods? Because there are a lot of methods who are based on assumption or on mass balances or something like that. But analytical methods are always useful for the evaluation of these estimations of emissions. They are valuable for the evaluation of your assumptions about the renewable part, which often are very wrong. So they are used for the cleanup of wrong assumptions. And of course, the analytical measurement should evaluate other methods which are maybe more cost effective for the counting emissions. And there are a lot of more special reasons why we need analytical methods. So in the waste area or waste community, the determination of the proportion of renewable raw materials is used for secondary fuels. We have heard about this recovery for liquid and for solid waste. It's done in the flue or exhaust gases which come out of the chimneys, direct or on adsorber. The analytical methods can be applied for the biogases itself, and of course also for the HVO and for other liquid mineral products. Analytical methods always need a quality control. There are accredited standards which can be accredited according to the highest quality standards for laboratory. And first of all, I like to mention the DIN and ISO 21644, which is for solid recovery fuels. And we also use it for liquid recovered fuels, not only for solid ones. It has historic reasons why it is just for solids, but it doesn't really matter. Then there's the other standard DIN 16640, which can be used for all sort of bio-based products. So it is used for food or for plastic. There is a lot of development for plastics with partly renewable raw material, and it can also be used for gases and things like that. More important is the standard DIN and ISO 13833, which is for stationary source emissions. So here we analyze the real CO2 emission, which is continuously sampled before it goes out of the chimneys. Many people always ask for the US standard, ASTM D6866. It's basically the same as the DIN 6640, but there's an important difference, as the US norm bases the result on the organic carbon of a sample, and the European norm bases on the total carbon of the product. If people work with the results, they really have to know this, especially if inorganic carbon is going into combustion and also goes out as CO2, because combustions also treat the inorganic carbon. Okay, I try to go a little bit more into these standards because they are slightly different, and later on I explain about the difference in the results. So, solid recovered fuels offers 4 different methods. The radiocarbon method using the liquid scintillation counting, the radiocarbon method using accelerator mass spectrometer, but also the method of selective resolution. It was widely applied and still is within the EU to determine the biogenic carbon content. It based on the dissolution in sulfuric acid. And the differentiation to radiocarbon method is that the selective resolution gives the biomass of a sample, while radiocarbon methods gives the percentage of biogenic carbon related to the total carbon of a sample. So the results are not the same. And the norm also describes the manual sorting in a qualified way. I'm not sure if it's still applied in some countries in the EU and if it can be used, but it's still there. So maybe people still use it. It's based on former methods where really a person stands at a table and was sorting a big bag of municipal waste. Second important or more important is the standard for the analysis of the flue gas. Both methods of radiocarbon method, liquid scintillation counting and accelerator mass spectrometer can be used to analyze the continuously sampled CO2 from the combustion oven. It is either adsorbed at some chemicals, at solid chemicals like soda lime, or it is absorbed at fluid soda. Both can be used and analyzed. I already explained about this DIN 16640, which we use for food additives. to differentiate between synthetic and natural additives. We use it for example, the analysis of beer and champagne, but it's also used for the HVO analysis or for biokerosene or things like that. Okay, now I will focus on the carbon-14 dating or carbon-14 analysis, the radiocarbon method, because it is a very elegant way to differentiate between fossil and renewable carbon. The method was developed in the 1940s in Chicago by Willard Libby. It is widely used in archaeology, where you probably all know about it, and in paleoenvironmental science to determine the age of various carbon-containing materials, wood, bones, whatever you think about. If it's in a certain time scale, you can determine it with radiocarbon if there is any organic carbon in it. We use it also for the determination of the age of groundwater, which is my main profession of what I do, and where our company was founded. Though since the 80s, my late boss has analyzed flue gas and analyzed food additives and things like that. So then there are special applications like dating of mortar or whatever you can think about. Then there is an important thing to differentiate between synthetic and biogenic products. And of course, what we are talking about is the determination of the biogenic content of CO2 emissions and or the combustion materials. What is the basis of the method? We have 3 different carbon isotopes, 2 stable ones. The main carbon is the carbon-12 with 98.89%. Then less common, the carbon-13, 1.11%. These stable carbons do not change in their amount of isotopes on the whole world. The third and less common is carbon-14, which is a radioactive isotope with a better decay, and it has a half-life of 5,730 years. The carbon-14 content in the sample is expressed in the unique percent modern carbon. Recent carbon of CO2 in the atmosphere now is about 100% modern carbon. And this unit means 30.56 carbon-14 decays per minute per gram carbon. It's based on an oxalic acid standards from 1953. Okay, why it is valuable to differentiate between biogenic and fossil carbon. Carbon-14 is produced in the high atmosphere by the reaction of nitrogen-14 with cosmic radiation. This is the only natural origin of carbon-14. The carbon-14 then reacts to oxygen and is in the atmosphere, of course. Any plant uses only CO2 from the atmosphere to build their molecules. Therefore, every carbon isotope in a plant comes from the atmosphere. So any plant today has 100% modern carbon. As all animals and we ourselves, of course, eat plants, all our carbon isotopes in our bodies are of 100% modern carbon. It might be different if you eat an old mammoth, but that's something different. There's also a total gas exchange with the ocean. So sediments and mussels and things like that in the oceans have also 100% modern carbon. Then the only decrease of carbon-14 in organic material is by radioactive decay. As we have such a high half-life of radiocarbon 14, it takes a long time to reduce this content, but it starts immediately. So if you die and we bury your body and we dig it out after 5,730 years, we will measure only 50% modern carbon. If we wait another 5,730 years and there are still some bones left, we will measure 25% modern carbon. After 60 years, we won't measure any carbon-14 in your bones. Here you see the decay curve. And now it comes. Any petroleum and natural gas which is used in the industry or used by human beings is more than 30 years old. So any petroleum, natural gas, or products from then are, as we say it, are dead of carbon-14. You can't measure any carbon-14 in these products. And therefore, it's a very elegant method to differentiate between synthetic and biogenic or bio-based material, to differentiate between fossil or renewable material. As anything which we produce today has 100, which we grow or built today is 100% modern carbon, and anything synthetic is 0% modern carbon. And this goes for any isotope in the material. But of course, it is not that easy. And now we come to the problems and uncertainties of the method. In the 1960s, we had a lot of overgrown nuclear weapon tests. And these tests increased the carbon-14 content of the atmosphere to more than 190% modern carbon. So we have a doubling. After that, we have a strong decline, which is mostly based on the CO2 exchange with the ocean. And the second effect is the input from the industry by fossil CO2, which is called the Suez effect. In 2022 or 2023, we are down or below the former 100% modern carbon in Schauinsland in Germany and Jungfrauloch in Switzerland. They measured in 2023 about 99.8% modern carbon. So this is the value which you should measure in any food which was grown in 2023. But if we go for wood or paper, this is mostly from older material. So if you think of a seedling in 1970 and a harvest of this tree in 1980, you may have a mixed result of 140% modern carbon in this wood. As waste, it has usually a high content of wood and cardboard or paper or whatever. We measure today higher results than actually is renewable. So we need correction factors for them. But as every tree and every paper production and whatever has a different time history, these correction factors are only an approach with significant uncertainty. The correction factors are defined in the standard. They are not defined by nations or by the EU, but they are given in this standard. For older demolition wood, it's a correction factor of 0.88, which bases on the mean value of 114% modern carbon. For wood material, it gives a higher correction, paper and cardboard lower. Fresh biomass doesn't need any significant, just a slow correction at the moment. And for mixed municipal waste, it's given a correction of 0.93. But you see, it does make a real difference. Here I've given you about 10 samples so that you see what it means. If I go for surge sludge, which is a typical value for a big city, then I have to correct the measurement value of the carbon 14 to result in the biogenic content related to the total content of the sample in percent. Then you see that it is about 1% less. This is the same for other materials who have no special correction factors. But if I go for demolition wood, you see that there is a very relevant decrease of the original measurement of 92.1 to 80.8%. Here are some other things. Of course, we can't measure any material which has no carbon in it. I don't know why we sent these samples, but anyway, if we go for mussels or beach sand, then you find also higher results, which is reasonable because they have a long life. Okay, this was the standard for solid covered pools. And now we go for the correction factors which should be applied to measurement of exhaust gases. We have to do a correction by the carbon 13 value, which is an analytical correction. It doesn't have much effect, but there is a correction factor given in the standard for 2011, and this correction can decrease for 0.3 according to the standard. So this year we do know any correction at all, and next year we will correct for 99.7% modern carbon. This is actually the yearly value of today. Sweden, that was very interesting, has different correction factors for exhaust gases. So if you have a combustion power plant in Sweden, you need to buy more CO2 certificates. Here are some examples for these corrections. I've given monthly samples of 3 different combustion companies. The first one is a typical municipal waste incineration, and you see that it goes down not really 1% with a correction factor. If you're in Sweden, you are far below, and it means a lot in case of money. The second incineration uses industrial waste. It has a far lower carbon 14 content. But you see, if the value is very low, you don't have much correction. The third is a power plant which uses demolition wood. And if you remember, if you analyze the demolition wood material itself, you have to correct it from about 97% to nearly 88%. If you use this standard and analyze the flow gas, then you don't have a decrease at all. So there is a real difference if you measure the sampling input or if you measure the sampling output. Both are applied within the whole EU. Some combustion plants measure the combustion materials, which goes into the oven, and the other measure which goes out of the oven. In the end, I like to explain something about the applications in the lab. So the combustion material, which has to be in fine grains and things like that, is combusted in the lab in a bomb to CO2 at about 600 degrees Celsius and about 180 bar, which causes a total combustion of the whole material. If CO2 flue gas samples, CO2 gas samples are used, which are adsorbed at some chemical, some solid or fluid chemical, then the release is done using phosphoric acid. If a pure gas sample should be analyzed, then it is combusted in a copper oven. All of this is handwork. No machines can do this. After we have produced the CO2, it has to undergo extensive cleaning because every other gas has to be separated from the CO2, especially sulfur hydrogen, as it all influences the measurement. So we have a lot of cryogenic things here to separate other gases. Now we have our pure CO2, and when we can decide if we want to use the method of liquid scintillation counting or if we want to use the accelerator mass spectrometry. For liquid scintillation counting, the purified CO2 is mixed with a scintillation cocktail. We need about 20 grams of CO2 for this sort of measurement. If we decide for the accelerator mass spectrometry, the purified CO2 is transferred to glass tubes and then graphitized. For this method, you need far less CO2, or you can't even process higher CO2 masses. And there you can see a difference between these two methods. So the method of liquid scintillation counting is more representative for a sample. On the other side, the accelerator mass spectrometry can give far lower uncertainties. You yourself can decide what you think is better. The result usually is the same, but the uncertainty is different, but also the representativeness. Okay, liquid scintillation counting. The sample mixed with liquid scintillation cocktail and put in a transparent bottle. The energy is given off by the radioactive decay of carbon-14. It's a beta emitter. And these beta particles are converted to light by this special cocktail and then produce a flash of light. This goes out of the transparent bottle. The light flashes are converted to electrical signals and amplified in photomultiplier. The recovery rate in our high precision counter is 74%. It's a low-level counter. Oh, we have different ones. And then it's just calibrated using standards. As the recovery rate is low, and as there are just a few carbon-14 isotopes in the samples who can actively react, we need a very, very well protection against the cosmic radiation. So in our lab, we have a bunker built vertically into the mountainside. And additionally, here you see lead stones which protect against all cosmic radiation going through the walls. Okay, the other method, so I come to the end, is the accelerator mass spectrometry. Here, the carbon isotopes are accelerated and the carbon-14 is separated by magnets from carbon-12 and carbon-13, so that only the carbon-14 goes into the detector, and then the particles are detected just as energy signals. And of course, this process is also calibrated by using standards. Okay, I guess I was very quick.
Yeah, very good.
Doesn't really matter. I hope now you have more insight in the analysis of fossil and renewable carbon, and that you understand more about the limitations, but also the possibilities of this method. Thank you very much.